Hi friends, this video is about plant breeding, its objectives and historical developments. This is very helpful for the BSc, MSc and the beginners who are interested in plant breeding. Coming to the definition of plant breeding, it's a science based on principles of genetics and cytogenetics. It aims at improving the genetic makeup of the crop plants. Different workers gave different definitions. The second definition is improved varieties are developed through plant breeding. Its objectives are to improve yield quality, disease resistance, drought, and frost tolerance, and important characteristics of the crops. Plant breeder has been crucial. The breeding is crucial in increasing production of crops to meet the even increasing demand for food. Some well-known achievements are development of semi-dwarf wheat and rice varieties, nobilization of Indian canes, and production of hybrid and composite varieties of maize, jawar, and bajra. Crop improvement means combining desirable characters in one plant and then multiplying it. The job of a plant breeder is to select plants with the desired characters, cross them, and then identify the offspring that combine the attributes of both parents. Then multiply the progeny to supply to farmers, growers, or planters. In modern era, plant breeding begins in the early part of 20th century after Mendel's work was rediscovered. Today, plant breeding is a specialized technology based on genetics. It is now clearly understood that within a given environment, crop improvement has to be achieved through superior heredity. The basic definition of plant breeding is the crop improvement as directed by humankind or as directed by evolution, and that is helpful in improvement of the mankind. The aim of plant breeding is to improve the characteristics of plants so that they become more desirable agronomically and economically. The specific objectives may vary greatly depending on the crops under consideration. Coming to nature of plant breeding, as we can say, plant breeding is an art, science, technology, as well as we can add anything. First one, the concept of art. In earlier days, man depends on his skills and judgment in selecting better plants. He knew nothing about the inheritance of characters, role of environment in producing them, and the basis of variation in various plant characters. The methods of selection was designed without the understanding of principles of inheritance. That means in the earlier day there was no link between genetics and plant breeding. Therefore, during primitive time, this breeding was largely an art and very less science was involved in that. Even today, success of selection depends upon ability of a person involved in the selection. That means selection is the basis of plant breeding. As a science, plant breeding is considered as the current phase of crop evolution. As the knowledge of genetics and other related science progresses, breeding becomes less art and more science. Especially discovery of Mendel's work or rediscovery of Mendel's work after 1900 added a lot of knowledge to science. Selection of desirable plants even today is an art, depends on the skill of a person. But alone skill is not enough. Modern breeding is a combined effort of art and understanding the use of genetic principles. Third, as a technology. Product of all breeding activities, whether dependent on the art or science, is improved variety, hybrid, synthetics and composites. This product is utilized by farmers for commercial cultivation. Therefore, breeding can be rightly viewed as a technology since it generates a useful product. The role of plant breeding. Human beings are dependent on the plants for food, shelter, clothing, fuel, etc. Drugs, entertainment are the basic things which humans are dependent on plant breeding. Now, the challenges before plant breeder. The first one is increasing population. Every 10 years the population increase like anything, and every second we can expect 7 to 8 children across the world. So to feed the increasing population with limited amount of resources and limited land, the increasing production and productivity is a major challenge before breeders. The second one is squeezing arable land. Day by day, the total arable land for agriculture is decreasing due to urbanization and industrial development. Breeders have to tackle this problem by realizing or releasing improved varieties of major crops, which gives better production per unit area. The third problem is erratic rainfall. This is a problem especially in the developing country like India, where majority of the cultivated area is dependent on the rain-fed agriculture and less on the irrigated land. So erratic rainfall causes major effect on the production. Fourth one is mechanization. The land fragmentation is a major problem to adopt the modern mechanization practices. So this is the biggest challenge before the breeders. Coming to the objectives of plant breeding. The prime aim of plant breeding is to improve the characters of plants that they become more useful automatically and economically. Some of the objectives are: first one is high yield. The yield in terms of grain, fodder, fiber, sugar, oil, etc. Developing hybrid varieties of jawar, maize, bajra helped in this way. Second one is improving the quality in terms of everything like size, shape, color, milling, baking quality, etc. The third is disease and pest resistance. The mutations, continuous mutation and evolution, directed evolution leads to continuous changes in disease and pest pattern in the crops. So considering this factor as a major challenge is very important. The fourth one is maturity duration. It permits new crop rotation and extend crop area. Thus, breeding for early maturing varieties suitable for different dates of planting. This enables the farmer to take 2 to 3 crops in a year. The other things are agronomic characters, photo and thermosensitivity, synchronized maturity, non-shattering characters. Non-shattering characters means shattering leads to losing most of the seeds, or we can say grain. Then determinate growth habit. Dormancy means overcoming the dormancy. Varieties for new season. New season means extending the cropping pattern to Rabi as or summer, or we can say Z period. Example, maize, which was only the Kharif crop and it became Rabi and summer also. That means making it to photo insensitive crop. Moisture stress, salt tolerance, that is nothing but the abiotic stress. Elimination of toxic substances, wider adaptability, useful for mechanical cultivation are the major goals in plant breeding. This is a gist or summarization of activities in plant breeding. First and foremost thing is creation of variation. How the variation is created? By the natural techniques like domestication, germplasm collection and introduction. These are the man-made or imposed processes in order to create the variation. The breeding processes to create variation are hybridization, mutation, polyploidy, so McLennel variation and genetic engineering. The processes which are given in the right side are maybe human made or occurred naturally. For example, we can't say the genetic engineer has happened because of human being. As we know, the basic or the natural genetic engineer Agrobacterium tumefaciens can do the genetic engineering efficiently comparing to human being, where the artificial techniques are still not popular comparing to Agrobacterium method. The creation of variation will be helpful for selection, then evaluation, multiplication, and distribution. To this, the supplements are creation of variability and naturally existing variability. Here this is just an explanation to the activities of plant breeding. Means this gives the importance of how the activities in plant breeding are helpful in the improvement of crops. Coming to scope of plant breeding, from time immemorial, the plant breeding has been helping mankind with knowledge of classical genetics. Number of varieties have been evolved in different crop plants. Since the population is increasing at alarming rate, there is a need to strengthen the food production, which is serious challenge to those scientists concerning with agriculture. Advances in molecular biology has sharpened the tools of breeders and brightened the prospects of confidence to serve the humanity. The application of biotechnology to field crops has already led to the field testing of genetically modified crop plants. Genetically engineered rice, maize, soybean, cotton, oilseed rape, sugar beet, and alfalfa cultivars are expected to be commercialized before the close of 20th century, but this could not happen. Genes from varied organisms may be expected to boost the performance of crops, especially with regard to the resistance to biotic and abiotic stress. In addition, crop plants are likely to be cultivated for recovery of valuable compounds like pharmaceutical products by genes introduced into them through genetic engineering. It may be pointed out that hirudin, an antithrombin protein, is already being produced from transgenic brassica apples. That means the genetic engineering and other editing technology are just a tool for modern plant breeding, but we cannot substitute them 100% for conventional breeding method. Coming to the historical prospects of plant breeding, the plant breeding history is divided into four era. The first one is pre-Mendelian, before 1900, Mendelian era, that is 1900 to 1920, post-Mendelian era is 1921 to 1950, and modern era is after 1950s. In pre-Mendelian era, in 9000 BC, first evidence of plant domestication in hills above Tigris River. In 5000 BC, agricultural communities existed in Mesopotamia. In 4000 BC, Egyptians used yeast to prepare wine and bread making. In 3000 BC, domestication complete for all important food crops in the Old World. In 1000 BC, this complete for all important food crops in New World. In 700 BC, Abyssinians and Babylonians hand-pollinated the date palm. In 1665, Hooke first described cell and knew as father of cell biology. In 1676, Millington, anthers function as male organs, he stated like that. In 1694, Camerarius first demonstrated sex in plants. In 1717, Fairchild gave first artificial hybrid, that is Sweet Williams into carnation. In 1753, Linnaeus, he published Species Plantarum, that is binomial nomenclature of plant taxonomy, officially begins with his general list of plant species. In 1761 to 66, Colleruter extensively studied the tobacco demonstrated that hybrid offspring received traits from both parents and were intermediate in most traits. In 1779, Knight emphasized the practical aspects of hybrids. In 1801, Lamarck gave the theory of evolution through inheritance of acquired characters. In 1819, Schirraff utilized pure line selection to develop a new oat cultivar and a new wheat cultivar. Oat cultivar released in 1824 and wheat in 1832. In 1831, Brown discovered eukaryotic cell nucleus. In 1837 and 1838, Scleeden and Swan developed cell theory. In 1859 to 89, Darwin published Origin of Species and noted inbreeding, sterility, and differences in reciprocal crosses. In 1866, that was a year breakthrough where Mendel published his experiments in plant hybridization, discovered unit factors or genes, law of segregation, law of independent assortment. He stated segregation in F2 and recombination of two or more genes and dominance of one allele over the other, formulated the law of inheritance. In 1884, Strasburger demonstrated fertilization and showed fusion of the two nuclei to form the zygote. In 1899, Navashin and Grignard discovered double fertilization of egg and endosperm. In 1899, Hopkins described ear-to-row selection method. Coming to the Mendelian era, as I stated, it is after 1900 when Correns of Germany, D. Rice of Holland, and von Sermack from Austria independently rediscovered Mendel's law of heredity. Bateson introduced the term allelomorph, homozygote, heterozygote, F1 and F2 in 1902. In 1902 only, G. Rice from Holland proposed mutation theory of evolution of experiments on Oenothera lamarckiana. Biffin of England, on his studies on inheritance of studies on disease resistance, found that Stiprus resistance was due to single gene. It was in 1905. In 1903, Johansen developed a pure line theory of selection. In 1904, Henning contributed for the idea of embryo culture. In 1906, Bateson introduced the term genetics. Yule gave the initial idea of multiple factor hypothesis. In 1908, Nelson Ehle gave an explanation for multiple factor hypothesis for grain color in wheat. In 1908 and 9, Hardy and Weinberg, Hardy from England and Weinberg from Germany, independently gave an algebraic equation that describes the within population, also known as Hardy-Weinberg law, that describes the gene and genotypic frequency variation in the population. 1914, Shell introduced the term heterosis. Blaxley discovered trisomics in the Datura. Coming to post-Mendelian era, in 1927, Muller reported artificial mutations in animals by X-rays. In 1928, Stadler described the mutagenic effect of X-rays in barley. In 1929, McClintock first reported and the 10 chromosomes in maize. In 1931, Stern, Crichton, and McClintock provided cytological proof of crossing over. In 1933, Rhodes discovered cytoplasmic male sterility in maize. In 1944, Avery McLeod McCarthy described the transforming principle and suggested that DNA, not the protein, is the hereditary material. In 1945, Hull proposed recurrent selection method. In 1946, Comstock and co-workers suggested reciprocal recurrent selection. In 1950, McClintock described the ACDS system, that is, transposable elements in maize. Some of the leftover scientists, Allard and Bradshaw in 1964 gave G into A interaction. Jenkins in 1940 gave recurrent selection for GCA, and Hull in 1945 recurrent selection for SCA. Davenport in 1908 gave the dominance hypothesis, whereas Floor in 1956 proposed gene for gene hypothesis. Harlan and Pope in 1922 backcross methods. Jones in 1914 double cross scheme. Jones and Davis in 1944 cytoplasmic genetic male sterility. Hopkins in 1908 ear to row method. Blaxley and Nebel in 1937 proposed colchicine term and discovered it. Golden in 1966 proposed SSD method. Lewis in 1954 self-incompatibility. Van der Planck in 1963 gave vertifolia effect. Vavilov in 1926 center of diversity and law of homologous series. Ashby in 1930 proposed greater initial capital hypothesis. Vilmorin in 1856 gave progeny test. Rimpu in 1891 found triticale. That was a successful distant hybridized crop. Then Hugo Dwerais in 1901 gave mutation term. Huggies and Babcock in 1950 sporophytic system of self-incompatibility. Nelson Nehle in 1909 bulk method. Karpanchenko in 1927 Raphinobrassica. Jones and Davis in 1944 male sterility. Young Long Ping in 1960 was the father of hybrid rice. Lewis in 1954 self-incompatibility classification. Painter in 1951 mechanisms of insect resistance. Atkins in 1953 modified bulk method. Jenkins in 1933 forage breeding. He practiced forage breeding. Then Burton in 1978, he is also a forage breeder. Chang in 1982 gave semi-dwarf rice varieties of Erie. He developed the IR series. Then Swaminathan in 1970, he was the grain revolution in India, as I already discussed. Orbach in 1949 chemical mutagen. Harlan in 1951 microcenter. Rhodes in 1950 male sterility in maize. Fisher in 1947 gave the components of genetic variance classification. Fisher actually gave in 1918. Coming to history of breeding in India. In 1871, Government of India created Department of Agriculture. In 1905, Empirical Agriculture Research Institute was established in Pusa, Bihar. In 1931, building of institute, that is IRI Institute, damaged in the ICR Institute damaged in earthquake. In 1936, shifted to New Delhi because of earthquake. In 1946, the name was changed to Empirical to Indian Agriculture Research Institute. From 1901 to 1905, major agriculture colleges were established at Kanpur, Pune, Sabur, Lyalpur and Coimbatore. In 1929, ICAR established, that was Imperial Council of Agriculture Research. In 1946, name was changed to Indian Council of Agriculture Research. In 1921, Indian Cotton Committee was established. Notable researchers on breeding and cultivation of cotton. Example, 70 improved varieties of cotton. In 1956, project for intensification of regional research on cotton, oilseed and millets, that is PIRCOM, was initiated to intensify research on these crops located at 17 different centers across the country. 1957, AICRP on maize started to exploit the heterosis, and this was the beginning for the exploitation of heterosis in different crops. In 1961, the first hybrid maize varieties were released by the project. ICR initiated coordinated projects for improvement of other crops. In 1960, the first agriculture university was established at Pantnagar, Nainital. In 1957, All India Coordinated Maize Improvement Project. This information is about the different hybrids. In 61, first maize hybrid was released, that is Ganga 1 North 1 Deccan and Ranjit. 1964, first sorghum hybrid, that is CSH1. 1965, first bajra hybrid HB1. 1991, first pigeon pea hybrid ICPH8 was released from ICRISAT, Hyderabad. Some of the eminent Indian plant breeders. Venkataraman was a eminent sugarcane breeder. He transferred thick stem and high sugar contents from tropical noble canes to North Indian canes. Saccharum barberry and officinarum were involved in this process. This is known as nobilization. The hybrid sugarcane became popular in India as well as the Western countries. Because of this, sugar production from sugarcane was improved and our country could stop importing sugar from other countries. He was honored by Queen of England in 1942 and given the title of Sir Venkataraman. B.P. Pal. He was during the period 1906 to 1989. He was eminent wheat breeder, developed superior disease-resistant varieties of wheat, NP varieties. First Indian director of IARI. Dr. Paul's major contribution is the Green Revolution, and he worked in the area of wheat genetics and breeding. He observed rust disease was largely responsible for low yield of wheat and therefore developed a systematic breeding method to develop varieties with resistance to rust disease. Swaminathan. He is responsible for Green Revolution in India. As I already told, he worked during 1970 and he is the initiator of Swaminathan Research Foundation in Chennai. He developed high-yielding varieties of wheat and rice, an Indian geneticist and international administrator. He is renowned for his leading role in India's Green Revolution, a program under which high-yield varieties of wheat and rice seedlings were planted in the fields of poor farmers. The other breeders. Pushkarnath was a potato breeder, N.G.P. Rao, sorghum, Ramayya, rice breeder, Ramdhan Singh, wheat breeder, Athwal, pearl millet, Bosi Sen, maize, Dharampal Singh, oilseed breeding, C.T. Patel, cotton breeder, who developed the world's first hybrid cotton in 1970 from Surat station, Shantaram, cotton breeder. Recently, R.R. Hanchinal, Rajiv Varshine, A.K. Singh, N.K. Singh and K.V. Prabhu are the eminent scientists. Thank you. I hope you liked this video. Do not forget to subscribe my channel. Thanks once again.